students, today I will give you an idea about the introduction to plant physiology and metabolism. So here the system is plant, right? And to describe the plant action, you have to know the metabolism, which ultimately describes its physiology as well as its other cellular functioning. 9 units are there, okay? So I will give you the glimpse or idea about each and every unit. Unit 1, plant-water relations. Unit 2, mineral nutrition. Unit 3, translocation in phloem. Unit 4, photosynthesis, very important thing for plant. Unit 5, respiration. Unit 6, enzyme. Unit 7, nitrogen metabolism. Unit 8, plant growth regulators, as well as Unit 9, plant response to light and temperature. Here you will know about the importance of water and pathway of water absorption, water potential and its components as well as its significance. Then you will know about the transpiration mechanism, significance, factors affecting transpiration as well as measurement, the ways by which the transpiration can be measured. Root pressure and guttation and its significance. Let's talk about them differentially. The first one, that is importance of water in plant metabolism, as well as the pathway of water absorption, water potential, its significance, components, etc. Here you have to know the study of importance of water in plants. So how far plant needs water for its metabolic purpose as well as for its physiological manifestation. To emphasize on the physical as well as chemical characters or properties of the water. To understand the physical forces that influence the water movement at the cellular level. To correlate physical factors with the water potential. Water potential is another very important factor or matter regarding plant-water relationship. To know about the cells and tissues, so how much it is responsible for absorption and the movement of the water through the plant or within the plant itself. Next, very important mechanism or process in case of plant, that is transpiration. The transpiration mechanism, significance and the factors affecting the transpiration and the measurement processes. So here you have to know, you can understand the transpiration and its different types. You can know the significance of transpiration. You can study the factors which are affecting the transpiration processes to know about the stomata, right? So the stomata, the opening and the closing processes can be regulated, and you also know the regulation processes as well. Understanding of different processes for the measurement of the rate of transpiration, and this rate of transpiration is very important for plant physiology to keep them cool. So you have to know all the processes as well as types. Next, another very important topic is root pressure and guttation and its significance. So what is root pressure, right? So root pressure is transverse osmotic pressure within the cells of the root system, right? That causes the sap to rise through a plant from stem to leaves. At night, in some plants, root pressure causes guttation. So what is guttation? That is exudation of drops of xylem sap from the tips of edges of leaves, okay? And this sort of guttation is actually observed in case of some grasses as well as in different types of fungi. Here you will study the nutrition of macroelements as well as microelements, okay? And transport of ions across cell membrane, the active transport as well as passive transport, the carrier, proteins, pumps, etc. The first one, that is macroelements, the source, function, deficiency, symptoms. So what are macroelements? Carbon, hydrogen, oxygen, sulfur, phosphorus, nitrogen, calcium and magnesium. So when we are talking about its source, obviously soil as well as water, right? We can get from there. And you have to know about their deficiency syndrome. So if they are deficient within the plant system, what sort of diseases can be happen? What are the symptoms? What are the syndromes? You will know about that. Once again, the microelements, right? You have to know the source, function, deficiency, symptoms. So what are the microelements? Iron, Fe, manganese, Mn, boron, B, molybdenum, Mo, copper, Cu, zinc, Zn, chlorine, Cl, as well as nickel. So we will know sources, function, as well as deficiency symptoms. If they are absent in sufficient amount within the plant system, so what will be the deficiency symptoms? As we know, some of the metals are acting as cofactors of some enzymes. If they are lacking, enzyme cannot support the plant metabolism, as well as that can be reflected in the plant physiology. Transport across cell membrane. So first you have to know the passive as well as facilitated transport, its mechanism, its kinetics, etc. Then there are some different types of ligand gated channels, ion channels, right, which are getting opened and closed by depending upon different types of signal, maybe stress, maybe light, maybe temperature, etc. And all those signals help to open the ion channels. So you will get to know about the carrier proteins, you will get to know about the pumps like sodium, potassium, ATPases, as well as you will know about the transporter and carriers and physiological function of all those pumps. So here two topics will be covered: phloem translocation, different theories including pressure flow, and the second one is composition of phloem sap, different types of experiment, and phloem loading and unloading. The first topic is phloem translocation, different theories including pressure flow. That is the conduction of food materials through phloem tissues. So here you will know actually the different types of phloem tissue, fine, and you will know all the functions of those phloem tissues and phloem elements as well. And you will know different theories which are involved in translocation of food in plant body. Now, the next one is composition of sap. So, first you will know the different types of translocation, maybe downward, upward or radial, as well as path of translocation. The blocking of phloem is very important. You know different types of experiments regarding ringing and girdling experiment, as well as mechanism of translocation, phloem loading, unloading, etc. So, all those topics will be covered here. Photosynthesis. First, you will get to know about the definition, site as well as chlorophyll. I will talk about that. Then you will know the photochemistry and accessory pigments. Then concept of photosystem, reaction center and cyclic photophosphorylation. You will know about non-cyclic photophosphorylation, oxygen evolving complex and quinone cycle as well. You will get to know about the electron transport and ATP production and details of C3 pathway. I will come to that. C1 pathway, C2 pathway, C4 pathway, CAM as well as photorespiration. The first one, photosynthesis. Definition, site as well as chlorophyll pigment. So we know that chlorophyll makes plant green. So it's a kind of pigment or the group of pigments which help to harvest energy from solar energy, right? So from solar energy, carbon dioxide and water is getting utilized to form starch or carbohydrate in case of plant by fixing carbon dioxide, okay? So the energy can be utilized, that food can be utilized by plant as well as for the heterotrophic all other organisms, by us even. Next, photochemistry and accessory pigments. So here you will understand the mechanism of photosynthesis, right? So first you will know what is photosynthesis, where the location is. Here you will get to know about the mechanism, the nature of the electron donors in the photosynthetic pathway, the pigments, the type of pigments, the main as well as accessory pigments, as well as photochemistry of the pigments, because the pigments are getting triggered or excited by a specific wavelength of light. Then only they become capable to transfer electron to other redox centers. So we will have to know about their specific photochemistry, on which particular wavelength the photon can excite them. Next, concept of photosystem, reaction center as well as cyclic photophosphorylation. This topic is also very interesting. So you will get to know about the concept of two system photosystems, the paths of photosystem as well, the reaction centers and its light harvesting complex or LHC. The role of light harvesting complex in state transitions, okay. So high energy to low energy state and different types of state. You will get to know about the definition as well as mechanism of cyclic photophosphorylation or CPP, non-cyclic photophosphorylation. Here you will know the Z scheme of photosynthesis, the mechanism of oxygen evolving complex, quinone cycle which occurs during the course of photosynthesis, the flow of electron in Z scheme of photosynthesis. One by one, the electrons are passing through different redox centers, as well as non-cyclic electron flow. Next, details of C3 pathway. The C3 pathway completes actually in 3 steps together. Number 1, carboxylation, number 2, reduction, and number 3, regeneration, right? So C3 plant reduces carbon dioxide directly into the chloroplast, right? And with the help of enzyme, ribulose biphosphate carboxylase, RUBPase, right? So 2 molecules of 3-phosphoglyceric acid is generated. As 3-carbon acid is generated, it is called C3 pathway. You will get to know about the detailed metabolism of this pathway. Next one, C1, C2, C4 and CAM as well as photorespiration, right? So contrary to photosynthesis, a respiration process is there by which by accumulating or by taking up oxygen, carbon dioxide can be evolved in the light, okay. So here we are having the different types of products are getting synthesized here. So during carbon dioxide fixation, when the photosynthetic plants produce 3-phosphoglyceric acid, it is coming via C3 pathway. But in case of photosynthetic plant, prior going to C3 pathway produces oxaloacetic acid, OAA, 4-carbon compound as their first stable product. It is called C4 or Hatch-Slack pathway, right? But when the plants absorb the energy of the sunlight at the daytime, they use this energy for the assimilation or fixing of carbon dioxide at night time also. This is called crassulacean acid metabolism or CAM. So here you get to know about the C1, C2, C4 pathway. So C4 named according to number of the carbon present in the oxaloacetic acid, the first product produced, as well as CAM. So all these are way by which the carbon dioxide are getting fixed. You will get to know about the anaerobic respiration, glycolysis and its significance, TCA cycle and electron transport system, pentose phosphate pathway and glyoxylate pathway, and some minor pathways including RQ values. The first one, anaerobic respiration. So here you will know how does anaerobic respiration work. You will know the characterization, application as well as importance of the anaerobic respiration. You will know about the ecological importance of anaerobic respiration as well. In case of glycolysis and its significance, you will get to know about the 10 sequential reaction steps involved in glycolysis. You will know the biological as well as clinical significance of glycolysis and the regulation of glycolysis as well. In case of tricarboxylic acid cycle and ETS, in this lecture, you will know about the location, significance, regulation and inhibitors of TCA cycle. You will know the reaction steps in TCA cycle. You will know the chemiosmotic mechanism of oxidative phosphorylation as well as redox centers or complexes involved in electron transport chain. PPP, pentose phosphate pathway and glyoxylate pathway. So in case of pentose phosphate pathway, that is also called hexose monophosphate shunt, you will get to know about the alternative pathway by which the oxidation of glucose happens. It is the alternative pathway of glycolysis as well as TCA cycle. So you will know the significance, sequence as well as outcome of this metabolic pathway here. The glyoxylate pathway is very specific, a variation of the TCA cycle anabolic pathway in case of some plants, microbes, bacteria, protists, fungi, etc. So here in this glyoxylate pathway, you will get to know how the acetyl-CoA is getting converted to succinate for the synthesis of carbohydrate. So these two specific pathway you will know here, the mechanism, the outcome, significance, etc. Reactions as well. The minor pathways. So all those are the major pathways, but there are some branching of the normal respiratory pathway where the alternative oxidases are utilized. You will get to know about their mechanism as well as reactions. Then RQ values, respiratory quotients. So these actually RQ values indicate which particular macromolecule is getting utilized, and they give different types of energy component. They are utilizing for different types of energy pathways. Say for example, in case of lipid, the RQ value is 0.7. In case of protein, it is 0.8, but for carbohydrate, it is 1. Enzymes, very interesting unit. So you will get to know about the classification as well as biological significance of enzyme, structure and properties of enzyme, different mechanism of enzyme catalysis, different mechanism of enzyme inhibition as well as kinetics, and the very important topic is allosteric enzyme, its principle as well as mechanism. The first one, classification and biological significance of enzyme. You will get to know about the classification and nomenclature, IUPAC nomenclature as well as EC number, enzyme commission number, according to the reaction it catalyzes. You will know the general significance, biological role, medical and industrial importance of the enzyme. You will know the enzyme units, control of enzyme reaction or activity as well as regulation, how the enzyme activity is getting regulated by different cellular factors. Then the structure and properties of enzyme. You will get to know about the three-dimensional structure, the catalytic triad, active site of the enzymes, different models of the active site, the general characteristics of the enzyme. You will know general as well as physicochemical properties of enzyme, the colloidal nature, reaction specificity, substrate specificity of the enzyme. You will know the structure as well as function relationship of the enzyme as well as the bioinformatics concept. Next, different mechanism of enzyme catalysis. So you will know the factors controlling the enzyme catalysis. You will know the different types of mechanism related to enzyme catalysis. You will know the enzyme purification as well as extraction process, grinding, purification, by which you will get the purified enzyme product. Next topic, different mechanism of enzyme inhibition as well as kinetics. You will know general mechanism of enzyme, kinetics, dynamics as well as reaction kinetic mechanism. You will know different types and mechanism of kinetics of enzyme inhibition, reversible, irreversible. You will know different aspects of single as well as bisubstrate enzymatic reaction. The last one, very important, allosteric enzyme, principle and mechanism. You will know the enzyme regulation may be done by the allosteric control, by covalent modification, by protein processing, or stimulation or inhibition by control proteins. You will get to know about the enzyme regulation by genetic control. You will know the principle, kinetics, mechanism, properties, models and examples of allosteric enzyme. Say for example, aspartate transcarbamylase. Here you will know the biological nitrogen fixation as an overview, molecular mechanism of NEF gene, NOD gene, nitrate and ammonia assimilation, and role of microbes in nitrogen, phosphorus, potassium utilization, NPK utilization. The first one, biological nitrogen fixation as an overview, the mechanism of biological nitrogen fixation. Here you will understand the mechanism of nitrogenase complex as a whole or its action. You will know about the NOD factors, NOD genes and their role in nodule formation. You will know about the NEF genes and their functions. The next one, the molecular mechanism of NEF gene, NOD gene as well as fixed genes. So, symbiotic nitrogen fixation genes in the broadest sense actually can be divided by NOD, NEF and fixed genes. So, NOD genes give rise to NOD factors, nodulation factors or NF. They are nothing but the signaling molecules produced by the soil bacteria known as rhizobia for the initiation of nodule formations on the root of legumes. The NEF genes are the genes encoding enzymes involved in the fixation of atmospheric nitrogen. The fixed gene represent the very heterogeneous kinds of genes which are related to development and growth in case of some metabolism of bacteroids. Nitrate and ammonia assimilation. Here we will understand the mechanism of nitrogen as well as ammonia assimilation in plants. You will get to know about the mechanism of nitrate assimilation in different microbes and plants. You will get to know about the illustrations of the mechanism of ammonia assimilation in bacteria and some actinomycetes. The role of microbes in NPK, nitrogen, phosphorus, potassium utilization. The microorganism living in the soil, they are actually the part of nutrient recycling, especially carbon, nitrogen, sulfur and phosphorus. Now these microbes decompose soil and improve its structure, subsequently improves the quality by increasing water holding capacity, infiltration, as well as it can enhance the quality of the soil. Thus, they are very essential for agriculture. You will get to know their importance here. Plant growth regulators. Different types of plant hormones, right? Like auxin, gibberellin, cytokinins. You will get to know about their classification, bioassay and functions. Different types of minor hormones like abscisic acid as well as ethylene. You will get to know about the practical applications of all those plant or phytohormones. The first one, auxin, classification, bioassay and function. So you will know the chemical nature of the auxin as well as according to the chemical nature their classification. You will know the development of the bioassays, how they are getting evolved as well. You will know the physiological functions of auxin and their major commercial or industrial application. Next, gibberellin, another very important plant hormone. You will get to know classification, bioassay and function. Gibberellins are the family of terpenoids compound made up of isoprene units, and it stimulates the stem growth in dwarf and rosette plants, as well as they are needed for sex determination, fruit production, etc. You will get to know the biosynthetic pathway and the factors that modulate the activity of the gibberellin as well as gibberellin level, active gibberellin level in plant. You will know the bioassay and applications. Cytokinin, classification, bioassay and function. You will get to know about the natural as well as synthetic cytokinin and which actually induce the crown gall, and you will get to know about the degradation of cytokinin in vivo. Cytokinin functioning as promoting shoot growth, chloroplast development, etc. You will get to know about the various uses and its bioassay. Next one, different types of minor hormones. Though minor hormone, they are very important. The first one is abscisic acid. So how they are getting synthesized in plastid and cytoplasm and how it is getting derived from zeaxanthin, another pigment. You will get to know about abscisic acid accumulation, homeostasis, and they are tightly controlled. Abscisic acid movement between organs and cells. You will get to know abscisic acid response, which are tightly regulated by other hormones, and you will know the uses and bioassay. Ethylene. Yes, ethylene is used as plant hormone in trace amount in case of plant. They are very much needed and they can stimulate plant to regulate the ripening of fruit, the opening of flowers and the abscission or shredding in leaves. You will get to know about its use, significance as well as bioassay. Now the practical application of phytohormones. There are many forms or many areas of agriculture like horticulture, pomiculture, moriculture, etc. In every aspect, there are uses of phytohormones. For successful cultivation and for greater yield, all those phytohormones are used. The high percentage of germination of sown seed on the field, and they will give the fruit bearing capacity as an output by using all those phytohormones. They are very much needed for tissue culture, in vitro plant genesis, right? So for somatic cell hybridization, so the derivation from plantlets, from the callus, introduction of recombinant DNA into protoplast, in all the cases, the plant hormones or phytohormones are there. Plant response to light and temperature. So this one is your last unit. You will get to know about the photoperiodism, introduction, definitions, different types of plants, then phytochromes and molecular mechanism of their interconversion. Photomorphogenesis, another topic and its application. The last very important topic is vernalization. Photoperiodism, introduction, as well as different types of plants, LDP, SDP, DNP. What are they? I am telling you. Photo means light and period means length of time. It is the reaction of plants and animals to the length of day and night. So how light can actually regulate the plant activity as well as metabolism. Most flowering plants have ability to sense changes of the seasons, okay? That means the length of the day and night as well as the flowering capacity can be regulated. To do this, they make use of photoreceptors. They are called phytochrome. They are nothing but light sensitive proteins, okay? They are actually excited by some specific wavelength of light and they can signally transduce that downwards. Plants need exposure to light for a critical duration and this critical duration varies from plant to plant. So, based on these critical durations, the plants are characterized into three categories. Number 1 LDP, that is long day plants. Number 2 SDP, that is short day plant, and number 3 day neutral plant. You will get to know about all of these in this lecture of photoperiodism. The second one, the phytochromes and the molecular mechanism of interconversion. So, phytochromes are very much sensitive to red or far red, AR or FR lights. So, there are some of the photoreceptors within the phytochromes, right. So, they have fundamental roles in photoperception of the light environment and subsequent adaptation for its growth and development. You will get to know its discovery, action modes of phytochromes and its general structure, their roles in seed germination, the chromophores, the two reversible forms of phytochromes as well as phytochrome gene family. Next one, very interesting topic, photomorphogenesis and its application. That I have already told, plant use phytochrome to detect and respond to red and far red wavelength of light. So, phytochromes are signaling proteins that promote photomorphogenesis in response to red and far red light. It absorbs red light and it will change to more biological active conformation, that is PFR form, right. So, you will know its application that is needed for the development of stage affected for seed germination, seedling development as well as photoperiodism. So, photomorphogenesis is actually coupled with the photoperiodism as well, that is sensing of day and night light and its morphogenesis towards the active conformation. So, first you will know what the photoperiodism, then how far it is related to the photomorphogenesis you will know. The last topic, very interesting topic, that is vernalization. So, it is the induction of a plant's flowering process by exposure to the prolonged cold of winter or by any other artificial equivalent. So, here I am talking about the cold or low temperature, some other stresses can be given also, and how far a plant is getting acclimatized with that particular environment. So, after vernalization, plants have acquired the ability to flower, right, but they may require additional seasonal cues or some weeks of growth before they will actually flower, right. So, not only these climatic conditions, these stresses, these factors are needed, but also they have to wait for proper season for flowering. This is sometimes used to refer the herbal flowery plants, non-woody plants requiring for cold dormancy to produce new shoots and leaves, but this usage is actually somehow discouraged or restricted, because something can be done in vitro, but we will always encourage a plant to respond spontaneously to its proper environment. So, that is why you will see its proper metabolism which can be manifested at its physiology. So, welcome students in this series of lecture where you will get to know about different types of plant metabolism as well as enzymes, plant growth regulators, plant hormones as well as mechanism of photoperiodism as well as vernalization, etc., by which you will get to know different aspect of botany. Thank you.